Chemical qualitative analysis is a way of separating individual ions not by how much is present, but by the specific characteristic reactions or solubilities. So the primary way we can determine whether an ion is present in a solution is by its specific solubilities, which are done in general by the group as listed in your lab manual, or by ion-specific solubilities that distinguish them from members of their own group, and by the color of either the original ion or the precipitate or solution that is formed during a characteristic reaction. Characteristic reactions are reactions with a specific compound that does something unusual that is not unlike other ions. For example, copper is often a light blue color in solution, but in the presence of ammonia, it turns a very dark blue, even at very dilute concentrations. It also will form a dark red precipitate when it is in solution with ferrocyanide. Qualitative analysis is a unique lab and different from the other labs we've done this semester in that there are no calculations involved. Qualitative analysis is the analysis of trying to determine what ions are present in a solution and not how much ions are present. There are many high-tech ways to do this, for example, X-ray fluorescence, but the importance of this lab is to teach some very important scientific skills. For example, attention to detail, making careful observations, and the ability to follow complex instructions. All of this will involve you using some critical thinking skills in order to determine which ions are present in your unknown solution. Before you proceed with the lab, it's important to keep in mind certain vocabulary that we will be using throughout the lab. When you have a solution, remember that a solution will be clear and that there will be no cloudiness. You will be able to see through the solution and there will not be particles in the way. It does not scatter light. This is different from being colorless. Something can be both colorless and clear, or it can be clear but have a color. Some of our solutions will be blue but clear, green but clear, or orange but clear. You will be looking for a precipitate which will appear initially as cloudiness in the solution, and then after you centrifuge as a solid that is collected at the bottom of the test tube, as you can see in the drawing on the right. The liquid that is not part of the precipitate, that is above the solid that is formed, is called the supernatant fluid. Sometimes we will need to filter our solution in order to separate the precipitate from the supernatant fluid. When we do this, the solid precipitate will remain in the funnel or the filter paper, and the supernatant fluid will go through the funnel to whatever collection vessel we have below, usually another test tube. Once it is collected, we call this the filtrate. This is the same thing as the supernatant fluid, it's just after it has been filtered. Sometimes we simply just want to pour off the supernatant fluid without filtering. We can do this because the centrifuge will make the solid precipitate form a very tightly packed solid at the bottom of the test tube, and generally this will stay in place. So if we pour carefully and slowly, we can decant or pour off our supernatant fluid into another test tube or into a waste container without moving the solid that is in the test tube. Before beginning your lab, you're going to want to read the directions in your lab manual. Read carefully about all the different techniques that you will be using and pay attention to the pre-lab considerations in the lab manual. You will notice that there is a description of the different groups and what kind of solubility patterns they have, which ions are in which group, and so forth. There's also a flow chart that will show you how we are going to separate the different ions. I have placed numbers next to each of these. These numbers will be labels for the test tubes that I am using. So I know that in my first test tube, I'm going to have all of the ions, and that only the silver in the form of silver chloride will form a precipitate. It will remain in the first test tube, and the supernatant fluid will move into the second test tube. I've labeled this so that I can easily go back and reference this when I want to know what is in a particular test tube. I have also labeled test tubes ahead of time so that I make sure that I have the right test tube for the right fluid. I have also prepared my lab manual. I've copied the flowchart into the lab manual along with the numbers. I've written down the names of the ions that will be used along with the observations of the color and the reagents that we will use along with the colors for them. Most of them are colorless, and so it is important to pay attention to the label. Then I have set up a table for Part 2 in order to make observations about each addition. I've left a little space so that I can make comments about how many drops I use or when I do an additional test or any kind of changes that I need to make. These are strips of red and blue litmus paper. If you've never used red or blue litmus paper, you'll want to pay attention to this video to show you how to interpret what you're seeing. Red litmus paper often appears a pink color, and blue litmus paper may be blue or even purple in color. Red litmus paper will tend to stay in the pinkish range if it is in the presence of an acid, but it will turn blue in the presence of a base. Blue litmus paper will stay blue in the presence of a base, but will turn red in the presence of an acid. In either case, blue means base and red means acid. I'm going to start by using KOH. Remember that this is a base, and I'm going to put it on this end of our strips. You can see that the pink of the litmus paper is turning to a blue color, while the blue litmus paper basically just looks wet. Now I'm going to do the same thing with an acid. In this case, 6 molar hydrochloric acid. I'll place this at the other end. The acid causes the pink litmus paper to return to its normal color, and it causes the blue litmus paper to become a darker red. In this way, you can know whether something has reached into the base range or the acid range. This test will be used to make sure that we have the proper conditions for our test tubes. One way to ensure that we are making this test properly is to stir the contents of our test tube before doing our test. This way we don't have a layer of an acid on top of a base or vice versa. These are the ions that we will be testing for in this portion of the lab. Notice that in some cases we have labeled with the actual chemical in the bottle, for example nickel nitrate, and sometimes just the ion. All of these are nitrates of the particular ions. Remember from your solubility rules that nitrates are always soluble, and this is the easiest way to make sure that when we combine these into one solution, the anion from one does not cause another to precipitate. Please take note of what these ions are and the color of each before proceeding on to doing your observations of the lab itself. These are the reagents we will use to test for the solubility and characteristic reactions of the different ions. Notice that most of them are colorless and all of them are clear. Please take note of the fact that they are colorless to begin with. Our one yellow one will be used in 2 different tests and will have a very different reaction between the 2 of them. Also pay attention as you pick up the bottles to what the concentration is, just in case there is something of a different concentration that is necessary for a different step.